This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 993, recorded on March 17th, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 4 degrees Celsius with a fresh breeze from the west. And in Fahrenheit, that's 37.
And I just saw Kathy in Atlanta for a TWiV we did at the APHL IDCon. I'm not going to tell you what those letters mean. You have to wait and see. But it was a good one. Very good. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It is 60 degrees Fahrenheit, 16 Celsius here, and very, very gray.
I think it's similar to what we have here. 14C, cloudy, yep. This looks like a little rain in the forecast. You have rain in yours?
I do have rain in mine.
All right. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 50 Fahrenheit, 10C. And I guess Kathy is now using the Beaufort wind scale, so I'll say we have a light breeze. Fresh breeze would be 15 to 20 knots, I think. And we've got west at about 5 knots.
I used whatever the Norwegian weather app had.
Ah, okay.
Let's see what they say about wind on the other app. 20 miles per hour.
Okay,
Gusts,
yeah, that's a fresh breeze, according to the
gusts
Beaufort
to
scale,
34. Okay. Cool.
So with 4 people, we have a nice squares, all symmetrical.
Yes.
On the video. It's
Time for Hollywood Squares.
There were a lot of them on those things, right? There
Yeah.
were more than 4. But the 4 is cool, so it's nice to have a big crew. That way, we can always have a good number of people. All right, before we do some science for you, Kathy has some announcement about ASV.
Yes. I want to start advancing beyond what we've been talking about. And part of my visit to Georgia was also site visiting the Classic Center again. And it's such a beautiful venue. You're going to love it. So I can tell you a little more about the facilities later, but I want to encourage everyone to start making their accommodations plan. If you want to stay in the dorms, you should know that there are buses that are going to circulate to the dorms, but they're also within walking distance if you're willing to walk 20, 25 minutes at most, probably. And the satellite meetings, I want to mention, there are 5 satellite meetings. You can find the details about them on the website. If you register for one, you can mix your visit to the satellites and go to the various other satellites that same day, which is on Saturday prior to the welcome reception. So it's sort of a pre-meeting. And so it costs $90 extra if you want to go to the satellites. But one is being organized by the counselors for virology trainees. And so they have a variety of people, diverse viruses. And so that's an exciting one. And then there's several other topics which you can check out on the program. So that's it for this week. But next time I'll tell you more fun things.
Fun things are always good. I have sad news.
Given how intrigued I am by some of the notes about these fun things on the show notes, I highly recommend you listen.
You should listen. You should always listen. I don't know why you wouldn't. Don't tell me it's 2 hours. You can 2x it or 1.5x it. It's just an excuse. It's an excuse.
So and the site is, of course, asv.org/ASV2023.
People who complain about 2 hours of TWiV think nothing of watching tennis for 4 hours or football for 8 hours or whatever. Do you think that's enriching your mind? I just lost 100 people. Yeah, I know. I gave up sports like 30 years ago because it was too much time to spend watching. When I was a kid, I used to like watching sports, but it's too long.
This might not be the weekend to say that.
What's this weekend?
This is the opening weekend of March Madness.
Yeah.
This is my big weekend.
Okay, everybody can do what they want. I just felt it was too much of a waste of time.
I don't watch March Madness, but I do watch tennis when there's a major tournament on.
I can always find the final score.
It's a requirement in our household.
We have sad news from an email from Eugenia, who wrote, Eugenia, one of the presenters on the mathematical TWiV 653 Back to School from August 14th, 2020, Janet Sinsheimer, has sadly passed away. She will be greatly mourned by her friends and longtime colleagues at UCLA, including those also on that podcast, Ken Lange, Mary Seal, and Alfonso Landeros. Janet was always an enthusiastic researcher and a kind mentor. She featured in many a pandemic Zoom meeting with the above and others. We've known her since grad student days. We'll miss her greatly. It's very sad. She was such a nice person on that podcast. You guys remember?
Mm-hmm.
Let's see, who was on there? It was me and Rich, so you guys don't remember.
Well, if we listened, we remember.
Yeah, it's too bad. Sorry about that. So she was related to the other Sinsheimer, right? Robert Sinsheimer, I think. Yeah. I don't remember how. Janet, an American expert in statistical genetics. Right. She graduated from Brown in 1979. Master's from Brandeis. Doesn't say anything about her Robert Sinsheimer, but I think I asked her and she said she was. It's a very unusual name, right?
Yes.
And she wrote, her dissertation was called, this is a great title, Extensions to Evolutionary Parsimony. Isn't that great?
That is good.
Concerning the
good.
phylogeny of evolutionary trees. That's so cool. I'm sorry that you have left us, but hopefully you enjoyed your time here, and many people will remember you. So thanks for letting us know, Eugenie. Appreciate it. All right. So continuing in our tradition last week, we explained everything. Let's do the same, folks, because people liked it, especially our cardiologist. He really liked that we explained everything. And so we will try to do the same, even though it takes us a bit longer. So we have a snippet for you, which was actually pointed out to me on the live stream over a week ago. Someone said, have you seen this? And I said, no, but it's amazing.
This is really, really
very
interesting.
I mean, for people who have been thinking about polio a lot, that includes Alan and I, and by a lot, I mean like a lot. Yeah, it's explanatory. It's published in Frontiers in Immunology. Case report. Clearance of longstanding immune deficiency-associated vaccine-derived poliovirus infection following remdesivir therapy for chronic SARS-CoV-2 infection.
And that's it. That's the...
Yes, but we will teach you.
We've talked previously about the...
Wait, let me do the authors. Hang on.
Sorry.
Birmingham, Canning, Wilton Kidd, Klapsa, Majumdar, Suri Kamur, Martin, and Huisoon from University Hospitals, Birmingham, Nottingham University Hospitals, NIBSC, which is like the FDA of the UK, and the UK Health Security Agency. Okay, what were you going to say, Alan?
Right. So I think we've previously explained the oral poliovirus vaccine. People should be familiar with this. You can go back to earlier episodes. Very effective, prevents disease, prevents infection, gives you great immunity. Downside is it can sometimes cause paralytic polio. The virus is a active attenuated vaccine, and so now the polio that's still in the world, is pretty much vaccine derived, right?
Yeah, and particularly type 2 derived. Yeah.
Yes. So one of the big problems is when we do these vaccination campaigns with oral polio vaccine, if somebody gets the vaccine and they have various types of immune problems, they can then shed infectious poliovirus for a really, really, really long time. And in this case report, they're talking about a guy who has apparently been followed for years for this, and they test his stool periodically, and he has been shedding poliovirus active poliovirus from the vaccine dose that he got as a child, and he's now 50 years old.
Yeah. So there are two kinds of circulating vaccine-derived viruses. They're the kind that you and I would excrete, called vaccine-derived polioviruses, and they're the ones causing the outbreaks in Africa, for example.
And that's just sort of a relatively short-term type of thing after you've been vaccinated.
Yeah, and
it's pretty much the same strain.
Up to 3 or 4 months. Well, it's not, it diverges quite a bit.
Okay.
It has over 1% sequence divergence from the parent. And the more it circulates, the more it diverges and becomes more wild-type-like. And they are neurovirulent. They can cause paralysis. So the guy in Rockland County here in New York who got polio because he wasn't immunized, it was OPV, vaccine-derived OPV2. But the immunocompromised people, they shed what are called immunodeficiency-associated
IVDPV with a small I, so it's like an Apple product.
These are people with severe combined immunodeficiency or hypogammaglobulinemia. So this SCID would be TB and what else, Brianne?
TB and NK cells are not working super well as well.
So between 1961 and 2019, there were 149 reported cases of these individuals who shed for more than 6 months. That's the definition. You're shedding for more than 6 months. And they will shed virulent virus that has been documented to cause outbreaks in under-immunized populations. And so they say no data exists to quantify the risk posed by them, and there's no strategy to manage them because mostly we don't know where they are because we don't check the sewage everywhere, so we have no idea where these individuals are.
When they are found and identified and they're in a place where they can actually potentially get treated, things have been tried to clear them of the virus. And that's why, Vincent and I look at this title and we're like, whoa, because people have tried various antivirals and monoclonal antibodies and polyclonal antibodies, and they talk in this paper about breast milk. They actually had this guy drink breast milk that was donated for a period of time,
it worked for a while.
It worked, it kind of worked for a while. But
then there was a discontinuity, and they couldn't get it anymore, and then it
came back. Yeah. So that's the problem here. You can imagine if we ever eradicated polio, which I think is not happening anytime soon. These individuals will continue to shed. We don't even know where they are. And if we could identify them, we have no way of treating them. And that's why, in part, the Gates Foundation has been funding research to develop 2 antivirals to treat these people, in part, for other things as well. And so that's why this paper is so exciting.
And now all of a sudden it seems like we've found one.
We have one. We didn't even try. But it just shows if you keep at something long enough, good things may happen.
Yeah, so this guy got SARS-CoV-2, and he was treated, and because he's immunodeficient, they, I guess, got right on it, and he got remdesivir, and what else did he? He got some other stuff too, didn't he?
Right, but the remdesivir was meant to treat the SARS-CoV-2.
Yes.
Yeah.
Not the polio.
Yeah. They had given up on treating this guy's poliovirus infection. That was just he secretes poliovirus. That's his thing. And then he got COVID, and they said, okay, well, yeah, he's high risk, and treat him with remdesivir, and so he went on it.
So he was 15 when he was diagnosed with common variable immune deficiency. So that's antibodies only, Brianne?
Yes. It's specifically a protein that's on B cells involved in B cell activation.
So these individuals have to be given immunoglobulin replacement. Otherwise, they get infections of various sorts. And so he was given these intramuscularly and then subcutaneously. And he's always being observed, right? So throughout his childhood and older, he's getting his vaccines. He got polio vaccine at 5, 7, and 12 months, boosters at 7 and 14 years of age. And in 1995, when he was 23, type 2 polio was isolated from stool. And so
he wasn't told until 1998.
Yeah.
that he
Yeah.
was shedding polio.
I don't know why. But, these are type 2 polio is eradicated. So he was a biohazard, essentially. And his stools were always sent to be checked, and they had to make sure they were sent by courier, really
Yes.
careful
Yeah.
and so forth. So no neurological symptoms. He never had paralysis, although the viruses that he shed were neurovirulent. And they did sequencing of all his isolates. There's a nice phylogenetic tree showing how his isolates all diverged, and they're consistent. The molecular clock, which means that the number of mutations this virus undergoes per year, was consistent with his last vaccination of OPV, causing it in 1986. And so what they tried was human breast milk, oral administration of normal human pooled immunoglobulin. You know what they have at the end of the paper? They have a statement by the patient.
Mhm.
Yes.
And he said the breast milk tasted horrible, but I
Yes.
kept at it just to see if it would work. You would think they would put something in it to flavor it, right? If it tastes that bad.
Guess.
I could think of things to put in it, like, some cherry or raspberry stuff.
I don't
but
know.
Okay. They gave you transient reductions in the virus in the stool, but never eliminated. There's a nice graph showing his stool virus going up and down. And the thing is that they did routine surveillance by cell culture, by isolation in cells, infectious virus, and also PCR, 27 stools, and with 4 to 5 times 10 to the 10th PFU, 1 times 10 to the 4th to 10 to the 5 infectious viruses per gram of stool. And then after November 2018, they didn't look at them anymore because it was too dangerous to mail the stool through the...
Yeah, I think it's in the patient statement. He mentions that the regulations kept getting more and more stringent, and it kept getting more and more inconvenient to get these stool samples processed. And so after 2018, they didn't keep at it.
Right.
But at that point, he'd been secreting this virus for 32 years.
Exactly. And after 32 years, you can sort of imagine that it's going to keep going.
You know where this is headed. Yeah.
So he actually was the subject of a paper a while ago where they documented this is the longest person known to excrete vaccine-derived polioviruses, but we haven't studied all that many. All right, so it's August 2021, he got COVID and symptomatic, test positive, went to the hospital because of his situation and got intravenous remdesivir for 12 days.
This is someone who, did they mention his vaccination status? Not that it matters.
COVID, I don't know, they don't mention it, no.
They don't, but could you have been vaccinated in August 21?
August of
Yeah,
20,
I think
he
so.
could have.
Yeah, you could have.
Yeah. Yeah, he would have been considered for early, one of the early cohorts.
Well, he lived in the UK, so we don't exactly know what their rules were. I know they had those bigger gaps between dose 1 and dose 2
Right.
and so forth, but it
But
seems
even
like
if,
he
I mean,
should have been
given the
prioritized.
nature of his condition, I'm not sure what it would have done. I mean, if you're lacking T and B,
Well, so he's not necessarily lacking T cells. He's just lacking antibodies.
okay, right.
Yeah.
Anyway,
So
so he got treated.
actually, the next paper is going to look at this in a different
Yes.
way. I don't know if they're lacking. I don't know if we look carefully enough to say they're lacking, right? Who knows? Anyway, so then he was in the hospital for 27 days. Oh my gosh, it's a long time for COVID, but I guess he's immunosuppressed. So he felt better. There was no more positivity, or at least the cycle threshold went high, and his symptoms were gone. Oh yeah, no SARS-CoV-2 on day 43.
Right. So
So
they
he
gave him he got a course of intravenous remdesivir from day
12
16
days.
of his illness.
And by the way, he did have a T cell response to spike and nucleocapsid protein.
Right.
So his T cells are okay. And he also had some spike antibody, but they say we don't know where that came from, right? It could have come from some of his immunoglobulin that he was getting.
Right.
But anyway, they decided to look for poliovirus in the stool, and it was negative.
Which was good thinking, right? I mean, they
don't know
gave up
why
in
they would-
2018 continuing following this guy, but hey, why not check?
Well, didn't he mention it in the patient statement?
Yes.
That he said something to the physicians about
that's right.
whether
That's
they
right.
should check?
Yes.
Well,
Yeah,
I even highlighted this.
Yeah, I researched online and was interested to read about affecting enteroviruses and whether it would have any effect on polio. Good thinking.
And then he called, that's right, the patient initiated this.
Yeah. And so and then they agreed. They said the immunologist excitedly brought to my attention. So they gave him, they checked, and it was negative. The first time they checked it was day 20th, the 5th day of remdesivir therapy was negative. And then after that, all the samples through August 2022 were negative for polio by PCR and virus isolation. So
they got it.
They took his last stool isolate from 2018.
The last positive one from 2018, yeah.
Before he went in for his COVID, and they tested it, and they said it was inhibited by remdesivir and polcapavir, another antiviral. But they don't show the data. They just say they inhibited 100 infectious doses of virus at 1 and 0.5 micromolar concentrations. And other antivirals they checked, ribavirin, favipiravir, and fluoxetine hydrochloride did not inhibit it. Did not inhibit poliovirus in cell culture. So all of that is, consistent with the remdesivir having eliminated his infection. So that is just cool. It's just cool that it's coincidentally. And so I would guess that, a number of people in his position were treated with remdesivir and may also have, if they had high VDPV, it would have gotten rid of it. So that's very interesting.
So my question for the two of you is, why do you think no one had thought of doing this before? Why would you have maybe not expected this?
I don't think you should have expected it wouldn't work. I think you should test it for sure.
I think it should have been tried.
Because remdesivir is a
it hits
rather
an RNA-directed RNA polymerase,
Yeah.
and polio's got one of those, and it's...
it's a rather broadly acting antiviral. It doesn't do everything, but it does inhibit other enteroviruses, non-polio entero. So, it's a matter of the people being in the right place. The people treating him didn't think of it, or they put two and two together. I don't know.
Just in general, why didn't virologists think of it? I mean, they were testing all kinds of drugs to possibly repurpose them, for SARS-CoV-2. Why not think about it in the other direction?
Because the number of brains thinking about polio were minuscule compared to SARS-CoV-2, right?
Right.
You always need a lot of people working on a problem.
Yeah.
And if you have 5, it's less likely to get solved. I mean, I didn't even think of it, and I'm thinking about polio all the time. I'm always thinking about these immunocompromised shedders, and it never occurred to me.
Well, I think part of it, I mean, at least from my perspective, I had read that people with VDPV had been put on antiviral therapies and nothing had worked, and I hadn't really delved into the literature to see what antiviral therapies they'd been put on, and I think the people putting them on the therapies are mostly doctors who are not specialized in this, and they're not necessarily talking to researchers in virology, and they're saying, well, what antivirals have we got? And, presumably they're coming up with plausible mechanisms and trying them out, but there's a limit to the number of times you can do this, and a lot of these drugs have side effects, and so there are just limitations on what got done, and this, wish we'd thought of it.
The remdesivir has been shown to inhibit other enteroviruses like Entero71, Coxsackie virus B3, Enterovirus 70, but nobody checked poliovirus, and I will tell you why they didn't, because it's hard to work on poliovirus now.
Yeah.
You can't work with types 2 and 3 any longer. They're eradicated, so you have to get rid of your stocks. That leaves you with type 1. And many places forbid you to work with any kind of poliovirus. They just want to get rid of it. So I think that's a dampener, right? But
Definitely
it's just
sounds like it.
Yeah.
It's just a great example of serendipity, right?
So the implication from this is that they ought to go back and look at all these other people who have this
Yeah.
long-term
Yeah.
polio.
Sure.
But what about thinking about using it just as an antiviral for unvaccinated people who get poliovirus infection?
So that's a good point. Daniel
if you
brought
know
that up.
early
Yeah.
enough.
It's got before if you do it at paralysis, you're gone. It's
Yeah.
too late.
Right.
Too
Yeah.
late, because that paralysis is a rather late sequelae, right? So you're already
Right.
past the initial viral replication phase. When you get paralyzed, the virus is in the CNS, and it's too late. It's the same thing with paralysis caused by enterovirus 68, right? In kids, by the time you see them, no antiviral or monoclonal is going to do any good. It's just too late. Although we should try, right? I think we get
Sure.
another person. I mean, what they do is they have some limb weakness, right? They go to their doctor, and then maybe a day later they go to the emergency room or whatever, and you should try it, but it's going to be hard to do.
Yeah, I wouldn't. The one thing I could think might be useful in that situation is, so yeah, give it to the person who shows up paralyzed, because it might help, and it's probably
Yeah.
not going to hurt. And I wouldn't hold out much hope there. But when that happens, that you probably have other people who might have not have been immunized or might not have been fully immunized, who are likely to be exposed in the area, and you're going to go in and try and vaccinate everybody. You could also distribute remdesivir to those same people, and try and contain any subsequent cases of paralysis.
Maybe only family contacts, because doesn't
Family
remdesivir have to be given IV, so
Yeah.
it's expensive.
Yeah,
Yeah.
it's not right.
Yep.
It's not something you're going to give out like candy, but it's something where people who were immediate contacts, you may want to.
Especially if they
this.
were maybe immunocompromised or something
Yes.
like that.
So what we should do next is to get these people who are identified as IVDPV shedders and do a trial
Yeah.
with them with this and see how it works on more than one patient. And then it becomes part of this eradication initiative, right? So if you identify someone, now you have something to do.
Yeah.
You can at least get rid of that from the environment.
Would it need to just be the immunocompromised shedders, or if you found someone who was shedding just because they weren't immunocompromised from having been vaccinated, could you also treat them too just to stop shedding?
Well, you do shed for 20 to 60 or 70 days, a couple of months. But maybe you need that to get a good immune response, right?
And I
just
think the
don't know.
problem is going to be that in the places where OPV is still being used, it's being used because the infrastructure isn't available to give people shots.
Yeah,
And if you don't have the infrastructure to deliver the inactivated, injected polio vaccine, then you don't have the infrastructure to deliver remdesivir either.
Yeah, I think that's probably correct. But I think it's still a great thing for these IVDPV shedders.
Yeah.
So anyway, there's a patient perspective, which is very cool. We mentioned, he said, in September, I learned the samples were negative for polio, which was amazing to hear. So he doesn't have to drink breast milk anymore or get IG immunoglobulin, which is good. I presume they will check him periodically, right, for poliovirus, and this maybe once a year, just to be sure that, because he is susceptible, I would assume, given his compromise, that he could encounter polio again, right, from someone else and get reinfected.
So I don't remember ever seeing one of these patient perspectives before, and I don't know how common they are, but I'm thinking that because he had something to do with getting them to test him, that they said, "Oh, it would be interesting to have your perspective."
Yes.
Or he said, "Could I write? Can I contribute or something?" And maybe that's how that happened.
Yeah, it may not be a regular thing. Yeah.
I think it's a great idea, though. I really like it. And one of the things that, case reports could benefit from is, what was it like to be that case?
Yeah.
So it's very cool. For polio aficionados, it's very cool. And I think it could really be helpful for the eradication effort, even though I
Yeah.
think the eradication will not work, but I still think we
Right.
ought to get rid of these shedders. By the way, there's an interesting side note here. Cross-reactivity has been demonstrated in poliovirus vaccinated sera. So you take someone who's been immunized with poliovirus, you take sera, and that sera will cross-react with SARS-CoV-2 RNA polymerase. Poliovirus vaccinated sera cross-reacts with SARS-CoV-2's RNA polymerase. So I have to look at that paper because, there are a number of caveats. So, but why would that be? I
Is
don't
this
know.
in people who have been vaccinated with oral or inactivated?
Now let's take a look.
Because oral, I could imagine, since you've got productive replication going on somewhere that is being expressed, you might actually have mounted some kind of a response from the polymerase being produced. I'm not sure how.
Antibodies to poliovirus and
Inactivated,
SARS-CoV-2.
I mean,
So these are sera from 204 adults and children who were immunized with polio vaccine. That's useful, folks.
any
Which-
particular polio vaccine?
Which polio vaccine? Study design, IPV.
IPV. Oh, okay.
And they found that antibodies to polio and SARS-CoV-2 were found in pediatric sera and from adults recently immunized with poliovirus. Sera from polio-immunized individuals inhibited SARS-CoV-2 infection of vero cells. So the problem here, folks, first of all, is that IPV should not induce antibodies to the poliovirus polymerase
Right.
because it's just a virus particle. There's no polymerase in it. Right?
Right.
Because they're saying here, these results suggest that the anti-polio polymerase antibody induced by polio vaccination may predict. No, there's
Do we know the inhibition in vero cells is due to antibodies against SARS-CoV-2 polymerase?
they have Western blots here.
Yeah, I mean, I guess you could theoretically do some sort of blast and see if there happens to be a cross-reactive set of amino acids, but
So
They have
so,
a Western blot.
we have-
Okay.
Go ahead.
Well, I think we have to go back a little bit for the jargon. So the IPV is the inactivated polio vaccine, and
Right.
you're saying that there's no polymerase there. There's no RNA-dependent RNA polymerase
In the particle. Yeah.
in the particle that's been inactivated.
Yep.
Right.
How do you know that there's no antigens left from inactivating the particle?
so there's no polymerase. You mean?
Well, there's
But
never
there's no
polymerase.
polymerase.
There's never polymerase to begin with. Right?
Oh, in the particle. Okay,
Yeah.
Right.
gotcha.
Poliovirus
Okay.
does not pack its own polymerase with
Right.
it because it has a positive
Right.
sense
Right.
RNA
No,
genome
I
that's
will
going to get
this.
translated
Yes.
to make the polymerase.
Yes.
There's a Western blot here, so I think we'll have to do this.
Yeah,
paper.
we'll have to do that paper
We'll have to
separately.
snippetize it because I don't want to spend more time because we have another very interesting paper
Yes.
which is important for us to talk about. So much for that one. Nature Communications, SARS-CoV-2 mRNA vaccines decouple antiviral immunity from humoral autoimmunity. And I made sure Brianne was on this episode before I picked it. So thank you for allowing us to do that here. This is coming from Yale School of Medicine, the Benya Roya Research Institute at Virginia Mason Medical Center. First, the authors, we have co-firsts. What do we have? Three co-firsts, yeah. Three first, Jillian Jaycox, Carolina Lucas, and Inci Yildirim. And then we have, do we have multiple senior authors? Yes, we have three. We have Kate Speak, Akiko Iwasaki, and Aaron Ring, all corresponding authors. So
So
go
before
ahead.
you get going too far, so the title I found not informative.
Exactly.
But so I'll repeat what the title is. SARS-CoV-2 mRNA vaccines decouple antiviral immunity from humoral immunity. And I found it very frustrating. And then I read the end of the introduction thinking, okay, they're going to give me the bottom line here, and they don't. So then I had to go to the final part of the paper where they tell us in conclusion what I think they should have had in the abstract. And then somewhere along the line, I'm trying to find it. Oh, a better title might have been something like at the end of the introduction about autoantibodies are stable during vaccination relative to their stability during acute COVID-19 disease.
Yeah, I didn't like to decouple. It didn't mean anything to me.
Yeah.
Right.
Yeah.
Yeah, I think decouple is just a really bad word to put in a title because then you're trying to think about a clutched, this is, okay, coupled, they were coupled? They were
Yeah.
what?
The title didn't make a lot of sense to me until I'd read the paper.
Yeah.
Yes,
And
for
until
sure.
I started thinking about their previous paper. And then I was like, oh, I get what this title means. But I sort of had to already know what the paper was about before I got what the title was.
Yeah, so I see where the title came from, but I don't think it was a good choice of title. Great paper.
But when
by
you
the way.
read it, you
Just
get
if
it.
the authors are listening, they're like, oh, God, they hated our paper. No, we like the
No,
paper.
the title
That's-
just is very
title
confusing.
does not sell it.
but if
and
you
the
do read
upshot
the
of
whole
this
thing-
for people who are trying to, well, what is the point of the paper? The real point of the paper is that the mRNA vaccines are not causing autoimmunity that you see when you get COVID infection.
Mhm.
So the vaccine is way safer than the infection. We knew that already, but this is another example of something that could have been a concern and is not.
Yeah, so they and others had previously shown that people who were infected with SARS-CoV-2 had these new autoantibodies.
Mhm.
And the
Antibodies
reason
against
that's
their own
bad
proteins,
is because
right?
that's your immune system responding against yourself. And
Yes.
there's
Right.
there's a whole library of diseases that are caused by your immune system responding against yourself, and you don't want any of them.
Yeah, and so, there was this worry that perhaps the vaccine could also do that. And
Right.
I can give all sorts of fun hypotheses and geek out about immunology about why the virus could potentially do this, but I'll hold myself back. But there still could be this question of, well, does the vaccine do the same thing? Are we, having some sort of risk here with the vaccine? And some people especially might think about that because they're like, well, we do seem to have a lot of side effects right after vaccination. There are people who have different, aches and fevers and being tired and things like that. So maybe we are inducing something that's not great.
And myocarditis, don't forget
Right,
that.
of
right?
course.
Right. And this
So-
would not be the first time something like this happened, that viruses or bacterial antigens can cause autoimmune responses. This is documented as something that can occur, right?
Yeah.
Yes. And they also mentioned that whether autoantibody responses after vaccination differ in people previously infected versus not previously infected has not been looked at. So they do that here as well. And the method...
I would say, so I think that's particularly important because people sometimes will say, oh, I was infected. Isn't that good enough? Should I also be
Sure,
vaccinated?
Sure. So the method they use is very interesting. It's called REAP. What's that song? Beware the Reaper? What is it?
Don't
Don't
fear the
fear.
Reaper.
Is that a Blue Oyster Cult
Blue
song?
Oyster Cult.
It is.
Yes.
Oh my gosh. They're from Long Island,
Are they? Okay.
Their only good song. They had one song and that was it.
We're going to get letters about that.
REAP. That's fine. Write to us. Get engaged, folks. REAP.
Rapid extracellular antibody profiling.
So they have, this is done in yeast, Saccharomyces, yeast, baker's yeast. This stuff is that you use in bread, right? Is that
Yep.
safe to
And
say?
beer.
That's
And
the
beer.
stuff.
And beer.
Yes.
So we can study these in the lab and modify them. And that what they did in this case is introduce DNAs encoding lots of antigens, 6,183 human extracellular proteins. That means proteins that are outside of the cell, right? Not inside.
I think they're all transmembrane.
Transmembrane?
Yes.
No secreted proteins. Okay.
Right.
Peptide
And the reason,
epitopes.
so these are things you can, this is called creating a library where you make a population of yeast cells in this case that each express one of these things. And then you can use standard microbiology techniques to separate them out so that you get a colony of yeast cells that express this protein, a colony of yeast cells that express another protein. And because they're all attached to the yeast, you get them nicely ordered on a plate. And you can then look and use this library to see what antibodies from a patient stick to what proteins.
Yeah, the way they do this is that the DNA that encodes this extracellular protein has a little bit of extra sequence. That's a barcode on it.
Right.
So the yeast will make this transmembrane protein. You can then bind that to antibodies and find out which yeast bound the antibodies. And then you do DNA sequencing on the yeast to see which barcode was pulled down.
Tells you what protein has been binding to that antibody. Right.
And since these are all normal human extracellular membrane proteins, they're things you should not be making antibodies against because that's you.
Yep. They also have thrown in there some SARS-CoV-2 epitopes, spike RBD, for example.
Yeah, to see if the vaccine made the antibodies it was supposed to.
Yes.
So that's the assay they use over and over to see. Again, you take sera from patients and you mix it with your yeast, making all of these different proteins on the surface of the yeast. Right, that's important. The antibody sticks to it. I think they pull out with magnetic beads at some point. Right. The yeast.
They pull it with beads. I don't remember if they were magnetic or not.
I thought I saw magnetic in the methods. But either way, let's say you can get out the yeast that are binding your antibodies, and then you do genome sequencing, and the barcode, as Brianne says, tells you what human or SARS-CoV-2 gene is in that particular one. So it's a good, quick way to figure out in a person what their antibodies are directed against, right? So that's what they use. They use this REAP library platform, and they say they give it a REAP score, okay, which is a reflection of binding, and they say that correlates with antibody titers. So they've done a calibration where they say, okay, if we get strong reactivity with the yeast display, does that correlate with antibody titers in serum? And it does. All right. All right, so the cohort here, these are important. So we have 3 separate cohorts, pre- and post-vaccination, groups of people who have volunteered. And this underscores the importance of having a cohort, not having friends, having a cohort of patients that you can go
not
to
co-hosts.
For pre-vaccine and post-vaccine and follow them up for a while. So we have one cohort, 33 healthcare workers from Yale New Haven Hospital. Half of them were seropositive for SARS-CoV-2. And the second cohort, 38 individuals with pre-existing autoimmune diseases and 25 matched healthy controls. These were from the Benya-Roya Research Institute.
And that's an important cohort because these people should have antibodies, or you expect them to have. Not that they should. You expect them to have autoantibodies.
And these are diverse.
They've been diagnosed.
They've been diagnosed.
They have different immunodeficiencies. Or different immune diseases, autoimmune diseases. We have 13 with MS, multiple sclerosis, 13 with rheumatoid arthritis, 3 each with systemic lupus erythematosus, type 1 diabetes, and Crohn's disease. So that's cohort 2. Cohort 3, 87 volunteers from the Dominican Republic who had previously received a 2-dose course of CoronaVac, which is an inactivated whole virus particle vaccine. And then they got a booster comprising the mRNA vaccine, which I believe was Pfizer here. That's what they had available. And then they have another group of 26 people that they monitored over time after vaccination, which comprised 14 healthy patients and 12 patients with type 1 diabetes. I know you're not going to remember all that, but just to let you know at the onset.
The final cohort is in the absence of vaccination, right?
Right.
Yeah.
So it's just to look at how many autoantibodies change over time.
Yep.
Right.
Yep. That last one was in the absence. Yep. Right. So you get sera from all these people. before and after vaccination, et cetera, and then you run them through your Reaper. All right, so everybody in the
You
healthcare
reap
worker
your data.
cohort were reactive to the SARS-CoV-2 RBD receptor binding domain by REAP after vaccination, which is good. They should
Good.
be, right? That's good.
And it's a nice change in the people who had been negative before vaccination, and in the people who were positive, they looked pretty similar
And
post-vaccination.
if you were infected,
Right.
then get a vaccine. It's either high or increased after vaccination. Yeah. Okay. Then this CoronaVac booster cohort, more variable, but mostly increased or remained stable, the anti-SARS-CoV-2 RBD antibodies. How about the autoimmune cohort? All the healthy people, most autoimmune people responded to vaccination. So they have antibodies to SARS-CoV-2 RBD. People who got anti-B cell depletion therapy, anti-CD20 antibodies, most of these are MS patients, had lower RBD antibodies after vaccination. That makes sense, right? You pull out their B cells, you get rid of their B cells with this antibody. And then they have, within this group, some patients taking ocrelizumab. Is that the anti-CD20?
I believe that is the anti-CD20.
They're less likely to make antibodies than patients with other autoimmune diseases taking rituximab, which is another. I know that rituximab is a CD20, right? Let's look it
I
up.
believe they both are, yeah.
Let's look up rituximab is used to treat blah, blah, blah. Come on. Tell us what it's against. Monoclonal antibody. Yeah, CD20.
And ocrelizumab is also a CD20.
All right. So,
And why would you be targeting CD20 in somebody?
So CD20 is on the surface of mature B cells. And in many of these patients, their autoimmune diseases are partially coming from making autoantibodies, making antibodies against a self-protein. And so if you could get rid of the B cells, you would stop making those antibodies. Unfortunately, we can't get rid of just the autoimmune antibody-making B cell. We have to get rid of all the B cells.
So
Which has side effects, but they're considered worth tolerating in these cases.
So MS in particular responds to anti-B cell therapy, for example, right?
It does.
Yeah.
Now, do these people, just a side note here, because the cardiologists might be interested, does treatment with these antibodies make you get more infections, or do you get some other kind of therapy to take care of it?
You are usually monitored closely for some types of infections. So you are immunocompromised.
Mm-hmm.
I'm less familiar with the anti-CD20 than I am with some of the anti-TNF antibodies, where patients are monitored very closely for tuberculosis positivity and certain parasite infections. But yes, you would be immunocompromised, and you might have additional infections.
Okay, so you have to be monitored, and
You have to be monitored.
Yeah.
I know of some people who are on some type of immunosuppressive medication for an autoimmune disease, and I recall them asking about getting vaccinated and how it would work for them. And so looking at panel D here, 1D, where many of these autoimmune disease patients who are being treated with a variety of things did seem to make lovely antibody responses, was interesting to see.
So they were lower. These patients made lower responses, and they say, in fact,
Some of them did.
most MS patients did not generate a SARS-CoV-2 RBD antibody response, but two of them, one of them not on any therapy and one taking dimethyl fumarate, made normal antibody responses. What is dimethyl fumarate? Another anti...
I think I am less certain
Let's look it up.
exactly. Let's see here.
Fumarate. Here we go. Is it methyl ester? I know. Psoriasis.
It just says that it decreases inflammation.
Yeah. All right, we'll get letters. It's fine. No problem. Anyway, those two made normal antibody response. But this is just to look at the baseline, what's going on with SARS-CoV-2. They also did other assays for antibodies outside of REAP. they looked for antibodies against the receptor binding domain of the spike of SARS-CoV-2 or the full spike by ELISA, which means just asking, do these sera have antibodies that bind to the protein? Or by neutralization assays, where they ask, do the sera have antibodies that will block infection of cells? And they found a good correlation between the REAP score, the ELISA titer, but not always to neutralization. So many of these patients that had these autoimmune patients, they make a normal response to vaccination measured by the REAP score or the ELISA. They don't have good neutralization titers, particularly the ones that are on anti-CD20 depletion. And so they say maybe the immunosuppression by anti-CD20 is skewing the kinds of antibodies that you're making. So they're not neutralizing. They're binding maybe somewhere which doesn't neutralize. It's quite interesting, right?
So at this point, we're at figure 1, E and F, and I was frustrated here because the x-axis of those had a bunch of abbreviations that I couldn't find.
Yes.
And I wondered whether maybe they were in the supplemental data or a figure that was originally in the main part that got moved to supplemental, but they were not explained.
Yeah, it took me a while to figure a lot of them out.
This is the way they put it, which I like. People on immunosuppressive therapy may make a constrained humoral response that leads to targeting of non-RBD epitopes. So antibodies against the receptor binding domain are largely the ones that block infection by SARS-CoV-2. It's the major target. And so what they're saying is that when you are doing this immunosuppressive therapy, you're still making spike antibodies, but they're no longer mostly directed against the RBD. They're elsewhere, and they're not neutralizing, which is very interesting, right?
Yeah.
It is.
All right, now, so that's virus antigens, okay? Now the real stuff, extracellular antigens. So they have these cohorts that, again, they're looking for antibodies against extracellular antigens by this REAP approach. The mean number of reactivities did not differ between controls or individuals with autoimmune disease, nor did they differ amongst the various autoimmune diagnoses. In this Yale healthcare worker cohort and the CoronaVac booster cohort, no difference in the number of pre-existing reactivities between those who had been previously infected and those who were naive. So vaccination is not changing the number of reactive antibodies against extracellular proteins. I mean, there are some, everybody has some, but they're not they're not different in these different groups.
Right.
And they found that the most autoantibody reactivities were stable over time in both vaccination and control cohorts. So those two populations, pretty much constant. So vaccination is not changing the reactivities compared to unvaccinated people. So presumably that's all a background that we all have of autoantibodies, and the vaccination doesn't increase that.
If anyone is looking at the data, I will tell you that if you're looking at figure 2, I found E to be the most useful panel.
Yeah. Okay. Let's see. So the dynamics of autoantibodies without vaccination were stable over time. In other words, you do multiple time points, you don't see a change. The variation is similar to the vaccination cohort. And the similarity between the autoimmune and healthy patients was still there even after you look at autoimmune patients on and off immunosuppression. So they say whatever new autoantibodies we see, it's not due to immunosuppression because we see the same results in both populations. All right, so now is the million-dollar question. Do the autoantibodies differ in vaccination versus people who get infected? So for this, they have this cohort of 36 moderate and 23 severe COVID patients who were hospitalized at Yale Hospital in 2020. They say the autoantibody trajectories, if you look at the REAP curves during vaccination, differ strikingly from those observed in patients with moderate or severe COVID who have a lot of increased and new autoantibody reactivities. So the autoantibodies they have go up, and they get new ones as well, new antibodies against self-proteins. And then during vaccination, they don't see this. They say it's exceedingly rare. Out of 1,034 total autoantibody reactivities detected among the vaccine cohorts, only 15, 1.45%, newly arose in the months after vaccination. And for COVID patients, they have 24 new autoantibody reactivities out of 463. That's just over 5%. And they say this is probably an underestimate because the library we used for the acute cohort was only 2,777 antigens compared to the other, which has 6,183. Right. Which I think it's a matter of timing. They did the other one before they had this updated
Before
library.
They had the better setup, yeah.
And by then, they didn't have enough material to redo it, I guess. Well,
right. And they point out that, they would really love it if they had pre-infection samples from those COVID patients. Sure. So they could do pre and post there. So they're only comparing kind of post-vaccine and post-infection here to see how many autoantibody specificities they have.
Right. But it's nonetheless, it looks pretty clear to me that the vaccine is really doing nothing to autoimmunity, and the virus looks like pretty bad news to autoimmunity, which is not a unique finding to this paper either. Their previous work and other papers have been telling us that COVID-19 infection is associated with some autoimmune response.
If someone's interested in that, their paragraph in the discussion is cited nicely with all of the relevant papers. I was like, oh, good. This is nicely organized for me.
We'll get to that. Yep. All right. So what factors are associated with increased autoantibody reactivities in patients who get infected, right? So what about the cohort? What they found was increasing age, being female, and clinical severity score of 6 were significantly associated with an elevated magnitude of increased autoantibody reactivities. So those are the factors that go along with having increased autoantibodies: age, female, sex, and clinical severity score of 6. Let's see. And conversely, changes during vaccination were similar to unvaccinated controls, like we said. No pattern in any particular person, age, sex, and so forth. There's no pattern available there. So it's just something that happens in everyone. And lastly, how about people with myocarditis?
Right. This is a big question because the mRNA vaccine is associated with a small but noticeable risk of inflammation of the heart muscle, myocarditis.
Yeah. Right. And so maybe there's a very small chance that the vaccine induced autoantibodies, and that led to some of the small number of myocarditis cases.
Right.
So they have 8 patients who presented to their hospital with MRI verified myocarditis that started 1 to 3 days after the second mRNA vaccine dose, and they all got Pfizer vaccine. They did not display elevated numbers of autoantibodies compared to controls. They did not have autoantibodies against cardiac or endothelial antigens or antigens that are known to be involved in cardiac inflammatory disorders. So it doesn't look like autoantibodies are involved in the myocarditis, right? However, they do say we are not looking at intracellular proteins, only membrane proteins. So they say we can't eliminate the possibility that there may be intracellularly targeted autoantibodies against myosin that could be involved in some of the pathologies. So that happens, right, Brianne? You can get...
That happens. You can get that. But they also point out some of the other data talking about increased frequencies of activated CD8 cells or NK cells that other people have seen in other studies. And in my mind, that seems more likely, but they certainly can't rule out the intracellular antibodies.
That's really that's the story there. It is.
I think the important thing, so you can't rule out that the vaccine is acting through an autoimmune mechanism to cause these rare cases of myocarditis. But based on the data here, the getting COVID-19 is way, way worse than getting the vaccine. And I think that's really the take-home message.
There's a measurable signal of autoimmunity with getting infected, and there's not a measurable signal getting the vaccine.
You could display some intracellular antigens, right? By fusing them to membrane proteins and so forth, if you really wanted to get at that. Anyway, so what makes these autoantibodies during infection, as Brianne alluded to earlier, that while they say inflammation and tissue damage and exposure of antigens that would be hidden, right? That could be. Here's one that I want to let Brianne explain. Okay, first, bystander activation of autoreactive B cell clones by a hyperinflammatory cytokine milieu. Oh my God, immunology, come on. What does that mean?
All right, so what that means is that we all have some B cells in our body that could make autoantibodies, but they have been energized or turned off. They've basically been told they're not allowed to make responses.
Okay.
And if they encounter extreme cytokine levels.
Okay. Like during an infection, right?
Like during an infection.
Okay.
Then they suddenly say, "Ooh, can I help?"
Yeah, exactly. Then suddenly they get turned back on, and they might start to participate where normally they are completely off.
Okay.
So the B cells that got turned off by some normal homeostasis mechanism might get mistakenly turned back on because there are just so many cytokines around.
Got it. All right. And then exaggerated proliferation of polyspecific B cell clones. So a B cell that makes more than one antibody?
Oh, I would think of it more as a B cell whose antibody can bind to multiple epitopes, maybe multiple epitopes that are sort of pretty similar.
So an epitope made against a virus that may be similar to a self-epitope.
Exactly.
Right? Okay. Those are their ideas, right? Okay.
And I will say the first idea is very much how I think about all of this.
Yeah, that's cool. They think that these are more likely than attributes of the spike leading to molecular mimicry. They think that these cytokine-mediated bystander effects are more likely to be the cause than, say, antibodies against spike reacting with one of our proteins. That's what they say.
Right. Well, because the vaccine would give you antibodies against spike, too.
Exactly.
So if that was likely, then they would see an opposite pattern in the REAP data.
Yeah. Okay. Now, here's an interesting paragraph. I like this. A lot of highlighting here. So they say CDC continues to recommend a third and fourth mRNA dose for immunocompromised individuals. And they say they're currently ongoing clinical trials. And they say, our study is limited, but highlights a limitation of relying on anti-SARS-CoV-2 titers as a correlate of protective immunity in immunosuppressed patients. Because, as you saw, we get binding antibodies, but they're not neutralizing. Right? Notably, we found that although most autoimmune patients generate RBD-specific antibodies after vaccination, they're non-neutralizing, unlike in healthy individuals, where you get a good correlation between antibody levels and neutralizing ability. So you can't just do an ELISA on immunosuppressed people. It's going to mislead you. They go, oh, yeah, you have plenty of antibodies by ELISA. Go home. No, they're not neutralizing, right? Am I reading their caveat correctly?
Yes.
So the reason for that skew in the antibody neutralization is very interesting, but they don't sort it out. And they also say we didn't measure T cell responses, which have been shown to be an important contributor to SARS-CoV-2 immunity. So they can't. They say we can't know the full degree of protection for these people because we didn't look at T cells. So I thought that was a good discussion there. And then finally, the last sentence: this work bolsters the safety profile of mRNA vaccines. Okay, folks, it's safe. They're safe. They don't cause autoimmunity, and highlights their ability to decouple. There's that word. SARS-CoV-2 immunity from the potential long-term autoimmune sequelae. I don't even
highlights
like
our
decouple.
ability to finally explain our title.
But I don't even like decouple in that context. Why can't they say this work highlights that the mRNA vaccines do not induce autoimmune sequelae or something like that, right?
Well, I mean,
they can't.
You can't really prove that from these data, right? And they already said there could be an autoimmune response against an internal cell protein that we didn't look for, and so forth.
I think really what they want to say is this work shows that the mRNA vaccine does not lead to broadened autoimmunity in the same way that infection does.
Yes.
And you think that they can shorten all that to one word, decouple. I don't agree with that. I think yours is better.
Yeah, I mean, the sentence before kind of says that there were no differences in the dynamics of autoantibodies against extracellular self-antigens, even in patients with preexisting autoimmune disease or vaccine-related myocarditis. So I like the first part of the paragraph better than that last sentence, which still has that word decouple as a shorthand, which I think is not important.
Maybe it highlights the ability of the vaccine to induce SARS-CoV-2 immunity without inducing autoimmune antibody responses.
Yeah,
yes.
That works.
Right.
Yeah, decouple, I think, is an inherently confusing word.
Mm-hmm.
Yeah. I mean, it has a certain meaning, but in a scientific context, it could mean a lot of things, right?
Yeah.
Right.
So,
because you immediately have to keep track of two things, and then you have to figure out what two things you're keeping track of.
Yeah.
And we're talking about the immune system here, so come on.
you know, I'm glad to see that all three of you had the same issue, because I said, am I stupid that I can't figure this out?
No.
You know, it's in a journal. It should be obvious to me, but it's just another example of how communication can be difficult.
For some reason, when I think about two things being decoupled, I think that it must be something about kinetics.
Yeah.
That could be. Multiple meetings.
Oftentimes when I am faced with, this is really making me feel stupid, sometimes in a seminar or sometimes reading a paper, when I go back and reflect on it, it's, no, I'm not stupid. It's that it wasn't communicated well to me, whether it was because some inherent problem I had with it or with the way it was presented by the presenter.
So one of the definitions is separate, right?
Yeah.
So SARS-CoV-2 mRNA vaccines separate antiviral immunity from humoral autoimmunity. Maybe it's a little better because they know what separate means, but anyway.
Yeah, I liked Brianne's reworking of it better.
Mm-hmm.
Anyway.
Okay, let's do some email here. Alan, can you take the first one?
Just a second. We find it. We're at Jason, right?
Yeah.
Dear TWiV, I think I can add a tiny bit more on why the Department of Energy might be releasing a report on SARS-CoV-2 origins. Rich was spot on, but here's a tiny bit more background. The Joint Genome Institute provides a link, the organization that coordinated the publicly funded wing of the Human Genome Project, is operated by the Lawrence Berkeley National Labs, which in turn is funded by the Department of Energy. It exists now as a user facility for advanced genomic research and continues to house a considerable deal of expertise across a wide range of biology. LBL also houses the Innovative Genomics Institute, whose staff and faculty include Nobel laureate Jennifer Doudna. It seems entirely reasonable that from a scientific standpoint, the JGI, Joint Genome Institute, would be a wing of the government weighing in on analysis of SARS-CoV-2 origins. Without an actual report with data, none of us can tell if this is a genuine, scientifically plausible report from experts qualified to weigh in, or if it's merely more speculation using the JGI as a cover to provide the report with an aura of science, or some third option. However, Department of Energy via the Joint Genome Institute would be a perfectly reasonable place to generate a scientific analysis, and until we see the report, there's certainly no call to question the appropriateness of Department of Energy to weigh in.
Okay. I think we said this more or less.
Yeah,
but,
that's all very good points.
But we need to see the data.
Yes.
And his P.S. Please forgive typos. I'm writing from a phone while walking my dog in the hills of Berkeley, directly above the Institute. And this doesn't lend to the best proofing.
Yes. Fair enough. I almost put this in as a snippet, by the way. There was just a report that came out analyzing additional data that was in a preprint put out by George Gao and some other folks. And then they pulled their data from the database, apparently, but it actually supports the market origin more.
Yeah.
So some of these SARS-CoV-2
specifically raccoon dogs
sequences from the market also had raccoon dog sequences in them.
Yeah. And it was in a preprint that the Chinese authors had put forth as showing that the SARS-CoV-2 sequences correlated with the sample that contained human sequences, suggesting humans there in the market. But apparently in that same graph, there was a data point that included an animal, and somebody outside of China analyzed the data, and it seems to be raccoon dog as a chief suspect. China, from the government level, has been trying to push the narrative that it came from entirely outside of China. Yeah, and so that's probably why the data supporting a market origin more got pulled, because they don't want it to be from the market either.
Nope.
But
that's right.
Doing that actually comes back to making it look more like maybe it was the lab, which you definitely aren't.
Yeah. They should rather have it be from the market than from the lab.
Yeah, but there's I'm sure a tremendous amount of pressure from the top, and I do mean the top, to say that it came from outside of China. They've got this theory that it came in on frozen food that was then sold at the market, and it didn't.
I think that they're really
they're shooting themselves in the foot.
They do not want it to be shown coming from the market, because they realize it shouldn't have been open.
Exactly.
And that they screwed the world by doing this.
I mean, okay, but go ahead and own that, because that's what happened, and it could finally, we need to get past this, maybe it came from the lab, which is not even, as somebody has pointed out, a hypothesis. It's just like there's nothing you can do.
Yeah. I saw an article that probably is the same article that you're mentioning all about this, and I was going through it looking very closely to see if there was a preprint or something linked, because I really wanted to read it and look at the data.
Well,
but there's a problem with the data now because
the for this new sequence that, by the way, it was on GISAID, and then they took it away once
they took it away.
Yep.
Yeah.
And so they're analyzing Eddie Holmes and the lady in France, I forgot her name, the epidemiologist, and Roby. They're all working on it, and they say they're going to publish something. So we'll see what they come up with. Their China's approach is don't admit anything, and it'll go away. Because they can.
That's only going to make it worse.
They can push people around.
It's the Streisand effect. I mean, you just.
What's the Streisand effect?
This is so. Barbara Streisand apparently tried to have an aerial picture of her house removed from a website, which of course caused that picture to multiply all over the internet and become much more known and shown than it would have been if she had just ignored it entirely. And so this has become known as the Streisand effect, where any effort to suppress information is only going to make it more interesting to people. Well,
it's a case of this sequence, right? As soon as the lady in France got interested, then China took it down, and she had already downloaded it, so now it's going to go everywhere.
Yes.
And them taking it down
makes it look like they've got something to hide, which is... Yeah.
All right, I'm going to take the next one about polio. Volker writes, I have been following your coverage on the active poliovirus found in U.S. sewage water. Can we call it infectious? Folks, infectious.
Yes, infectious. Okay.
And I have a question that I hope you can help me with. Despite the constant inflow of people from regions where OPV is used and the majority of people in the U.S. being immunized by IPV, why is there only a small amount of poliovirus found in sewage water, which in several cases can even be traced to one spreader? My assumption is that vaccine-derived poliovirus should be everywhere, as almost everybody's guts can be infected. I would like to understand this better. From Bavaria. Okay, here's my take. First of all, we're hardly looking anywhere for poliovirus in sewage, right? We looked at a bunch of places in New York and a few other places, hardly at all. So I don't think you can say there's only a small amount of poliovirus found because we haven't looked enough.
Well, there is only a small amount found. But you can't say there's only a small amount there.
Yeah, because they're not looking very much. And also, I think the importation of OPV is sporadic. I think it comes in, it spreads, and then it fizzles out, and then it comes in again. And if we had been looking over the previous 15 years, we would know these dynamics, but we decided not to look.
Could this be partially related to something that you mentioned earlier, that the shedding happens sort of for 3 or 4 months after after vaccination? So the only people who would be shedding are those who happen to come in 3 or 4 months after they've been vaccinated?
Well, yes, but also it doesn't have to be someone who's just vaccinated, right? They could be infected by a vaccinated person and then bring it in. They're going to shed it for. So it could be either one.
Okay.
But in either case, if it's a vaccinated person who got infected or if it's somebody who got OPV overseas and then came to the U.S., unless they're immunocompromised, it's going to stop after a little while. Yeah. And unless they spread it more widely in their community, it's just going to peter out.
Now, I do advocate a wider surveillance system for poliovirus. Yeah. I understand that poliovirus is a containment issue now. I get it, because types 2 and 3 are eradicated. So you don't want anyone just handling sewage and potentially infecting other people. I get it. So develop a protocol that will inactivate it shortly after sampling in some way, right?
Well, I mean, if you're handling sewage, you should have infection containment protocols and not specifically for polio, because it's sewage. It's got everything in it. It's
the problem is there's a polio containment authority now that you have to and they make their own protocols. They're different. Yeah. And if you don't follow them, they yell at you.
But we should be setting up screening systems in any country that can afford to, which would certainly include the richest one on earth. We should have screening of sewage as a routine thing where we test for a whole panel of things, right? And we found during the pandemic, this is a really good way to monitor viruses in the community.
So just so you folks know, part of CDC is the U.S. National Authority for Containment of Poliovirus, the NAC. And they have a website, and they have, all sorts of information about the inventory and containment, what you should be doing. And if you want to work on polio, which some people need to still work on it for making vaccines and diagnostics and so forth, and for doing wastewater surveillance, you have to answer to the NAC. It's like kissing the ring of the Pope.
I want to be sure that we address Volker's statement where he says, my assumption is that vaccine-derived poliovirus should be everywhere, as almost everybody's guts can be infected. So the second part is true. Almost everybody's guts can be infected. Mm-hmm. But the vaccine-derived poliovirus has to be from sources of people who got immunized by the oral poliovirus vaccine.
That's right.
And that's not everywhere. And so that's why the vaccine-derived poliovirus is not everywhere.
Well, it has to be brought in from countries that are using it, right?
Correct.
And but you could imagine scenarios where it's brought in and it circulates for extended periods in the U.S., depending on hand hygiene, right?
Yeah.
I mean, we're having an outbreak of norovirus now, you could easily have poliovirus piggybacked onto it, and you wouldn't know because there'd be no disease, right? So,
Right.
Yes, I agree. We don't know we don't know where it is at all because we haven't done.
We're not
surveillance.
looking.
We're not looking, and that's why we should have been looking. Don't you want to know where it is? Like every state, and now I'm told, oh, it's expensive, and the CDC doesn't want every state sampling because of this containment. The NAC doesn't want people sampling so I think you should work it out because it's really important information. Good question, Volker.
It's the whole thing about what do you want to spend your money on? Do you want to spend your money on the military being prepared to fight wars in 2 continents or whatever that thing is?
Yeah, sure.
Or do you want to spend it on surveillance and pandemic preparedness and things like
Which
that?
Which is a different kind of war.
Yes.
Exactly.
Too bad. So we have to make that choice.
But for polio monitoring, as I say, I think the way to get this implemented would be to put it as part of a comprehensive monitoring program. And yeah, fine, put the protocols in to handle polio on the assumption it's going to be there, but monitor a bunch of things. Don't just look at one. Anyway.
So we have 2 emails about female jazz musicians. Maybe we should save these for Dixon.
Probably save those for Dixon.
All right, so I'll take those out and move them. So that brings us to John.
No, Patrick. No?
Patrick. Wait.
Patrick was
right.
another female jazz singer.
Okay.
No,
so
John.
John, yeah.
John.
So Kathy, could you take John's, please?
Dr. TWiV, it would not surprise me if Paul Berg's classic protein synthesis video in the dance idiom had already been a pick, but if not, it should be. You, of course, know the video, but even better, someone has recently dug into the history of it, which is here along with an embedded link to the video, and Paul Berg even lived just long enough to comment on it. Roger Hendricks was at Stanford at the time, but when I asked him once if he knew any of the background about it, he didn't, so here it is now. So here's a link about protein synthesis, an epic on the cellular level. Otherwise, the wind woke me up a couple of times last night here in Greater Braddock, but by the afternoon it had become a relatively civilized and sunny 7C. So I had fun listening to this being discussed, the video, because I used to have my genetics students watch it, and things pop into my mind. 30S ribosome, peptide bond. So, yeah. Anyway, it was fun to hear about, and it will be fun to take a look at this background. Thanks, John.
Yeah, it's the making of. Cool.
Yeah.
And I think this was Rich's pick last time
Yes.
or something
Yes, it
like
was.
that.
Mm-hmm.
Right. Brianne.
And we
Sorry.
there was just a lovely obituary of Paul Berg because we just lost him in February, right?
Mm-hmm.
Yeah.
And David Baltimore has, I think it's in Nature. He wrote a very nice tribute to Paul Berg.
Yeah, I think
That's
we
just came
we
out.
did mention it last week, right?
A different obit.
Different obit, but yeah, the fact that he passed, yeah.
Right.
Brianne.
Patrick writes, Dear TWiV, just listened to the latest podcast and just wanted to say that as an idiot layperson, you're not an idiot.
No.
I am so appreciative that you define the terms and explain the acronyms. I know it must be a pain for you to do that, but for someone like me, it makes all the difference in the world. I would be totally lost if you didn't take the extra time to explain the basics. I learned so much from listening to TWiV. For example, I was also wondering if they pool plasma to get more diversification or just use a single patient source, and you answered that for me and so many other great questions. Thank you very much, Patrick.
Always glad to be of service. Let's do another round, Alan.
Bill writes,
Did
Hi,
I
TWiV
read
team.
any?
What?
Yeah.
Oh, did Kathy?
You read the Berg obituary.
Sorry, you're right.
Okay.
You can read another one. I'm sorry.
In the next round. Sorry.
Okay. Bill writes, Hi, TWiV team. There's a disagreement on the origins of SARS-CoV-2. So what? Let the experts pull their hair out trying to figure out what actually happened. The next spillover is coming. The magnitude of the next spillover depends on our actions. Here are some suggestions to cover our butts, regardless if it was a lab or wild spillover. You are welcome to add your own suggestions. 1. The study of existing pathogens and novel potential pathogens, along with their reservoirs and vectors, to gain an understanding of how they could possibly spill over into human populations. 2. International agreements to provide unbiased teams to study potential spillover links to help prevent future spillovers. 3. The study of novel antibiotics and antivirals, to the stage where safe and effective versions can be quickly manufactured and released when needed. 4. Changes in antiviral, antibiotic, and vaccine design and production so they may reach all human populations without delay. Shouldn't our representatives be working on tangible solutions rather than pointing fingers? All the best, Bill.
Good luck.
Working on tangible solutions rather than pointing fingers is a wish a lot of us have had about particularly the American government for a very, very, very long time. I would add consistently funding public health at the state and local as well as federal level in some mechanism that would last longer than an election cycle would be a great, great measure to implement.
Yes, working on the public health infrastructure is always very helpful. I give my emerging infectious disease students a reading that was written in, I believe, 2018 that talks about some issues with the public health infrastructure, and it is honestly infuriating to read now because you can shake your head and see all of the things that people were talking about beforehand that could have helped.
Yeah.
I would add to this something that is being promoted by Ian Lipkin and the Center for Infection Immunity called GAP, the Global Alliance for Preventing Pandemics. And what they're trying to do is establish infrastructure in key zones where we're talking about laboratories to do screening throughout Africa and Asia. And they will be at places where there's a lot of human movement, train stations, bus stations, ports, and so forth. And they will regularly be taking samples. So maybe you would get an early warning of something new or novel coming up. And so this program, GAP, the goal is to build the capacity in these countries. In other words, build the labs, train people to do it, and then leave them be so that local people have the jobs and they do it, and then have it.
But keep funding them.
Yeah, exactly.
Keep funding them. But he's trying to raise money to establish them initially. And then eventually, you have to keep funding them. But I think it's a good idea rather than looking at all the bats in the world and trying to figure out what might come. I mean, you have to, as he says here, Bill says, studying the pathogens in their reservoirs. I think that's good to continue to do. But I really think having key places to do diagnostics would help as well. I like that idea.
Yeah, I think back to the BAT meeting and that talk from Raina Plowright about sort of ecology changes that can impact spillover frequency and how much we could actually think about some of our behaviors that might reduce the chances of spillover. So yeah. I would put that in that first bin, and that's a big one for me.
I also like what you mentioned. Ian Lipkin is promoting the idea of looking at surveilling people as opposed to surveilling the animals.
Exactly. Yeah.
Yeah, we're increasingly understanding that spillovers are not rare events that once they occur, you get this pandemic. They're things that occur repeatedly, and eventually you get a particular virus may spill over into people dozens of times, and then finally it gets into somebody and manages to get established in a way that transmits to somebody else, and that's the key event. But if you'd been looking all along, and you'd seen those previous dozens of spillovers, you would have said, hey, this is something we ought to keep an eye on, this virus.
Yep. So
you may not have to go to the bats. You may be able to see it when it's coming into the humans beforehand.
Kathy, can you take the next one?
Bo writes, hello, TWiV team. I just listened to Epitope 987. Very informative as always, but it seems there's a little confusion between plasma and serum. Basically, like Dr. Casadoval said, plasma is the liquid portion of your blood. Serum is the liquid portion without the coagulation factors, so it is collected after coagulation has happened. If you spin the blood right after collection, you'll get plasma. If you let it sit for a while, coagulation will happen for most people, and you will get mainly serum after spinning. Or if you let it sit for a while, even without centrifugation, you will see a layer of somewhat clear liquid, depending on how much lipoproteins and whatnot are suspended in there. There would be serum, that would be serum too. In practice, blood is drawn into different types of tubes. If the tube is made of glass or has a silica coating, which will enhance coagulation, after 1 hour you can spin and get serum. If the tube is coated with anticoagulants, for example, heparin, citrate, or EDTA, these two are chelators that can take out calcium, which is important for the coagulation cascade. Coagulation factors will remain soluble, and you will get plasma after spinning. Plasma donation uses a mechanism that separates plasma and pumps back your red blood cells right away. Anticoagulants are also involved, I think some form of heparin. Because of silica in serum collection, it wouldn't be a good idea to use sera to treat patients. Coagulation also activates the complement system like crazy. The first time I wrote I was a research scientist in academia. Now I work at a local health department's public health lab. We do wastewater SARS-CoV-2 testing, so I got curious how RNA levels could be so high in wastewater and consulted a former colleague. They developed a feline COVID model, and I am a cat person. I was told that it's not uncommon for viremia to happen with COVID, and the virus in the bloodstream can infect epithelial cells in the intestines with high ACE2 expression, and people can shed intact virions. I did find a few studies on finding coronavirus-like particles in the blood and culturable virus in fecal specimens. And Bo gives a couple of references, coronavirus-like particles in plasma and viable virus in fecal samples. Second time writing, first time donating. Became a sustaining member just now. I'd also like to advocate for careers in public health. Graduates, please check out your local health department for jobs in labs, clinics, or epidemiology departments. So first of all, thanks, Bo, for donating and becoming a sustaining member. That's much appreciated. And stay tuned for the episode that Vincent and I did,
Right.
where we talked to people about how they got to their careers in public health labs and in clinical labs.
Yep.
It's a whole arena of careers that we don't hear enough about in biology and microbiology, that these people said basically once they figured it out, they couldn't go back. There was nothing else they wanted to do, and they all seemed really happy with their careers. So, yes.
I think, Bo, the major source of SARS-CoV-2 particles in feces is from you swallowing mucus. That's a normal way of getting rid of particles from the respiratory tract. There's a mucociliary elevator that brings it up out of the lungs. You swallow it and out it goes. You see this with many respiratory viruses. And I think that's where most of the viral RNA comes from in the wastewater. Brianne, can you take the next one?
Sure. Rohit writes, "Hi guys, this is Rohit. I'm an immunology student at Caltech. I'm curious if you could talk a bit about whether there are heuristics to decide a priori if animal testing is a good indicator of human prediction, and what you do if it isn't. I was reading about how certain aspects of autoimmunity don't replicate in other species, and it seems like a very tough problem. Best regards, Rohit." It's a very tough problem is a great way of stating it. I don't know of any specific ways to come up with a heuristic for this. There are certainly certain types of diseases that match certain animal models better or worse than others, but we don't know why that is necessarily, other than sometimes something about the physiology of a particular organ in that animal versus in humans, but it's not something that we can, have an algorithm for or something like that.
Okay, remember last week's discussion of 1918 influenza in non-human primates.
Yep.
There's no heuristic. Nope. You would think a primate would be a good model, right? It's not. Looks like
you basically you test it out and see what the symptoms look like, and oh gosh, this looks a lot like the human disease, so we're going to use this as a model. Or, well, this kind of looks a little like the human disease, and it's a mouse, so it's cheap, so we're going to use this as a model. It's not not a clear decision point. One of the many reasons that animal models are limited,
Right.
and they're just they're the least bad way of approaching the problem
Right.
in a lot of cases.
When we're trying to make these animal models, it's because we want to answer some question about a disease that we don't currently know the answer to. And so if we don't know everything there is to know about the disease, how can we say that the animal version is the same?
Right.
Looks like we have a class at Caltech emailing us.
Mm-hmm.
This is Chris. Hello, I'm an undergraduate at Caltech, currently taking immunology, BI, biology 114. I wanted to thank you for putting on your show. I often find it difficult to really know which immunology papers are groundbreaking, considering how much background is often required to understand them. Your podcast makes it a lot easier to understand and digest the findings in these papers. I wanted to ask if there may be a way to induce specific memory B cells against certain antigens in a vaccine immune response. As was discussed, the intention of Scott's work, I guess that's Scott Hensley, is in part to create a vaccine that forms the basis of future immune responses to vaccine when given in the future. I'm curious about what other cofactors, if any, might be necessary to ensure that the future vaccine-caused responses are strong enough against an injected antigen to ensure that memory B cells are activated and undergo proliferation. I look forward to hearing more about this topic. I have no idea. Brianne?
So from my understanding of the question, and I think there are a couple ways I could read this question, but from how I'm looking at it, Chris is asking about whether one could use a vaccine to kind of push memory B cell production uniquely, or to really get mostly memory B cells as opposed to plasma cells or something like that. And I think that there are people who have ideas about doing that by either changing cytokines that are delivered with the antigen or using adjuvants to get you certain cytokines. But there's not anything that is actually something that we're using. These are more ideas that I've seen people working on research-wise.
All right, let's do some picks of the week. Brianne, what do you have for us?
Speaking of memory B cells,
I figured this was going to be your pick.
there was an XKCD comic earlier this week called Lymphocytes and Their Functions that is really funny. It describes a number of different types of lymphocytes, gives you a little drawing and tells you what the cells do, starting with plasma B cells that turn out antibodies, but then going to naive B cells that try to stop pathogens by asking nicely, or memory B cells that quietly sing memory from cats at all times,
Yes.
and on and on and on to some not real types of cells and some real types of cells, and it's just really funny. It made me laugh. A whole bunch of people sent it to me, and I laughed every time.
Yep.
Oh, so to get the alt text, you click on the alt text, right?
Yes.
Yeah.
That's
And
funny.
the alt text is good.
Yes, the alt text is also quite good.
Cool.
It's a very good XKCD.
Kathy, what do you have for us?
Uh-oh, I was looking at the XKCD.
Hey.
Oh, yes. I picked an article that was sent to me by Justin, A Brief History of Parafilm, and within it is a video. And the first time I read it, I thought, this didn't give me as much history or as much background about parafilm as I wanted, but I think I was trying to skim it that first time. So watching it or reading it the second time and then watching the video by somebody who's not the author, I found very entertaining. So the video talks about how, it starts out by stretching some and telling what we use it for. And the article fills in a lot of gaps about the difference between paraffin and parafilm. And I just really enjoyed it. So
The video is really good.
Yeah. Is it really, does it really keep water from going through? And in fact, no water vapor goes through parafilm. So stuff will dry out, even if you've parafilm wrapped it. And what is paraffin? Well, it's the wax that forms on top of cooled petroleum and so forth and so on. So I highly recommend it, but definitely check out the video.
I'm very excited to watch this because parafilm is one of my favorite lab supplies.
Yes.
I love
Parafilm is great.
Love it. Parafilm is awesome. Yep.
And one thing I learned in the video is you can apparently get it in wide rolls.
Yes, yes.
Which I may actually have to get for the kitchen because...
I have wanted parafilm for the kitchen for years.
What are you going to do with it, Alan?
There
just
is...
any place I would use plastic wrap. I mean, it's...
There is that
better.
wrap that's sort of like parafilm, but it's made for the kitchen.
Yeah.
Have you not used that?
Yes, that's a good
I
one.
Yes.
don't think I've tried that one.
No,
Okay.
I want parafilm in my kitchen.
I want parafilm.
Right.
Yeah, the one Kathy's talking about is a little more papery like. It's crinklier, but parafilm is softer, yeah.
And it has a sidedness to it. One side is more sticky than the other, which I
Okay.
didn't notice for a long time. But
Parafilm is so cool. Whenever I used it, I always thought this is such a great secret of labs,
yes,
right?
yes. And the video is by this person who has done a series of things called "What Is This Thing?" And then I happened to click on, at the end it leads to a bunch of them, and there's the last one is "What Is This Place? The Lab?" And I only just started watching it, but she's talking about the fact that she's not going to have a lab anymore. So I left that for later.
Alan, what do you have for us?
I have something that's been in my picks for a while now, but this is an underwater exploration article and video. So this organization, the Global Foundation for Ocean Exploration, they're a nonprofit based in Mystic, Connecticut, which is a neat place to visit, by the way. They've got a cool maritime museum. But these folks do ocean exploration and especially shipwreck exploration, and this is their... all the stuff on their website is really cool, but this is their exploration of the USS Monitor, which is an extremely important historic site. It transformed naval warfare. This was the first modern battleship, and it was constructed in the Civil War in like 3 months. They threw this thing together because they heard the Confederates were armoring a ship, and so they put together this steamship with a rotating turret with cannons on it, and they got into a fight with the Confederate armored ship and fought to a draw. And then, the war proceeded. But then while the Monitor was being towed to another location, it sank in a storm, ended up on the bottom just off Cape Hatteras. And what's cool about this video is, first of all, it's this historic shipwreck, and this has now been designated a marine sanctuary to keep people from looting it. But they're down there with an ROV, remotely operated vehicle, showing footage of the wreck. The wreck itself is kind of cool, but what's really interesting is how much life is on it, because it's right off Hatteras, and there's a tremendous amount of marine life in that area. And you'll see in the video sand tiger sharks circling. They're the first thing you see, but then all these schools of fish and this tremendous amount of marine growth on it, and the whole thing is turned into this tremendous artificial reef down there under the water. Just very, very cool to watch.
They used to put old New York City subway cars out off the coast to make reefs.
I've dived on those.
You have, really?
So I've dived on artificial and what we would call natural wrecks. There's a tremendous collection of these off the East Coast. There are some that are just piles of like concrete pillars that form a reef structure. There are some that are barges. The old subway cars, piles of those. There was I did one dive on it was a collection of like these armored vehicles from the Vietnam War era that they'd sunk and turned into a reef. One of my dive buddies had been in the Army, and he said, "Yeah, that's the best place for these."
Hopefully they take all the oil out of them, right?
Yes, so there's a whole process where they decontaminate these and get all the fossil fuels out, and when they're ships, they pull out the asbestos that was used as insulation and all that. But the ones that ended up there in a storm or got sunk by U-boats, they just went to the bottom.
For other trivia aficionados, the Monitor fought against the Merrimack. I had to Google to make sure I was right on that.
Yes, the Merrimack was originally the USS Virginia, and the Confederates captured it and then hacked it. Basically, they threw a bunch of armor on it and turned it into a steam battleship of sorts, but it wasn't as sophisticated as the Monitor, which had the rotating turret.
Gosh. All right. Thank you. My pick, I think we have picked before, but I wanted to pick it again. It's Skype a Scientist, the creation of Sarah McAnulty. We just talked about her paper on TWiV yesterday, and she's a squid biologist. And the squid that she works on, the bobtail squid, they're cool because at night, they're feeding up by the water, and so that the predators don't see them, they glow. So they mix in with the moonlight. And the glowing is done by bacteria, by Vibrio, which and in the daytime they go in the sand and they get rid of the bacteria, and then before they leave for the night, they get the bacteria back. And it's just so cool. But it turns out there's another organ that depends on bacteria, and that's what Sarah works on. So we did her paper yesterday, and we had her send in comments, and she said, please plug Skype a Scientist. So I thought I would do it here. And this is a organization, it's a nonprofit that connects scientists with schools and other people
Operators
out there
are standing by
who want to have you go talk to their group, and they will do it. It's usually one-on-one. And they think, I think this is great because they think, as she said yesterday, that we think conversations are the best way to teach, which of course we do too here on TWiV. But this has got a unique approach, so you should check it out. And if your organization wants to try it, just go to their website.
And are they still Skyping a scientist, or are they Zooming a scientist at this point?
That's a good question. I don't know. I have encountered people who still use Skype. I remember we used to use it all the time before COVID.
That used to be the way we did TWiV.
And now it seems kind of clunky when every now and then I go on some call on Skype, and it's very clunky. But I don't know. I guess they still are but I actually don't know. It's a good question.
On the other hand, I'm starting to feel that way about Zoom now that we're doing Riverside, and I've started doing Riverside for my interviews for work, and it's just.
But Skype a Scientist
the next thing.
Rolls off the tongue, where Zoom.
Yes, it does.
Zoom or
Riverside a Scientist doesn't quite
doesn't
have
work.
No,
doesn't work.
No.
I mean, that's when I do the clinical update with Daniel. I say, joining me tonight from New York, and last night he was in Arizona. I said, joining me tonight from Arizona. It just doesn't roll off the tongue in the same way. None of the states do. Well, or, live from Arizona, TWiV. That doesn't work.
Yeah.
So anyway, skypeascientist.com. Check it out. And you can engage them if you would like. And that is it for TWiV 993. Show notes are at microbe.tv/TWiV. You can send us questions and comments to twiv@microbe.tv. And if you enjoy our work, we would love your support. It doesn't have to be a huge amount. It can be like a coffee a month. And you can go to microbe.tv/contribute for different ways that you can do that. Let's see. So we had to take Amazon Smile off the page because they don't do that anymore, which is too bad. But we have Patreon, we have PayPal, we have Venmo, and we have checks. Every week I get a few checks from you lovely folks here at the incubator. So there are ways to do all of them. And we are a nonprofit, so your donations are U.S. federal tax deductible. They go to a good cause, which is educating people about science. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker, Drew University Bio Prof. Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here. I learned a lot.
Alan Dove is at alandove.com and theturbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV. It's viral.